In your study guide handout for chapter 23, you should have a couple of different eukaryotic supergroup phylogenies. They are from a previous textbook and they should generally show the same groups, but some of the organization might be just a little bit different. The phylogenies are set up and printed just a little bit differently, but all of the groups should be represented here in this phylogeny and in your study guide packet. You will notice if you look at your study guide packet, there are a couple of additional labels that phylogeny shows, and that's okay. The addition is that, or one of the additions, is that in this land plant lineage here, we have a name in our lecture, and the name of this lineage is the Archaeaplastida. I don't know why our textbook doesn't use that name. It just doesn't. The other description that you'll see is the SAR label. SAR stands for the first letter of each of these groups. The Stramenopila, the Alveolata, and the Rhizaria. They are sometimes classified into their own group and just generally called the SAR group. The other thing you'll notice is that the study guide packet that you have puts Amoebozoa and Epithecanta into their own monophyletic group based on shared ancestry. I am not super worried about the fact that this one doesn't show that. It doesn't really matter too much. A lot of the origins of these protist supergroups are going to be a little bit cloudy. Some of the relationships maybe haven't even been fully figured out yet. For example, the relationship between the Alveolata and the Stramenopila is a little cloudy. They're not really sure where the common ancestor was, which group the common ancestor might have been more closely related to, etc. So the remaining videos for the protist section are going to cover each of these groups. There'll be a couple in each of the videos. And then after that, we'll move into fungi. So at one time, protists were classified as their own kingdom. So we had fungi, animals, plants, and protist kingdom, along with a single bacterial kingdom. Archaea used to be lumped in with the bacteria. Protists, all of them used to be their own group, and then our multicellular organisms used to be their own group. But protists don't form a monophyletic group. What that means, it should say eukaryotes don't form a monophyletic group. There are some multicellular organisms that come out of the Archaeaplastida lineage and also the Epithecanta lineage. So eukaryotes have some characteristics that overlap. Multicellularity is just one of them. For example, you might see some organisms that are photosynthetic in the Excavata and in the Alveolata, but there's also photosynthetic organisms in the plant lineage. You may see some organisms that move like amoebas in the other groups. For example, there are pseudopod structures that are similar in Rhizaria to the Amoebozoa. So we see a lot of characteristics pop up multiple times in these groups. So we have many characteristics that are not monophyletic. And then, of course, we've got that whole multicellularity problem. Multicellularity is not monophyletic either. Multicellularity evolved 3 different times within this lineage. So it evolved out of the land plant and relative lineage, and it evolved 2 different times out of the Epithecanta lineage. So we are commonly going to see structures called plastids. Plastids are basically just pigment-containing organelles. Generally, those organelles are photosynthetic, although in some cases they may not be, but they are generally going to have a pigment inside that gives them a particular color. Most of them are going to be photosynthetic. Plastids are a general term for that structure. The most common example that you know of is the chloroplast. The chloroplast is a plastid whose pigment molecule is chlorophyll. The plastids arose in eukaryotes by primary endocytosis. We talked about this already in chapter 22. We'll review it here again. And primary endocytosis took place when a cyanobacteria was brought in by phagocytosis, and a symbiotic relationship was formed between an archaeal cell or cell of archaeal ancestry and a chloroplast. Many of our protists are photosynthetic, but they are photosynthetic because of endocytosis, not because they evolved from a cyanobacteria. They evolved as an archaeal group. Eukaryotic traits began to form, such as invagination and enveloping the DNA, and then that acquisition of the cyanobacteria occurred. Just a very quick review. Primary endocytosis is the process by which we get both the mitochondria and the chloroplasts. I'm not going to say too much about this here because we already have this in our notes. You can certainly pause it and write down anything you would like from this slide. But the main way that we know a plastid was derived from endosymbiosis as opposed to the entire cell being photosynthetic like a bacterium is that it has a double membrane. So the first membrane comes from the membrane of the cyanobacterium itself. So there's a plasma membrane around the cyanobacteria. And the second membrane comes from the organism pulling in that plastid via phagocytosis. So phagocytosis is where a vesicle forms and that plastid gets pulled in to the cell. Now the cell membrane that enclosed that organism then wraps around the membrane of the organism itself. So we have two, one that was the organism's originally and one that derives from the envelope that surrounds that cell as it's taken in as food. So this leads to single-celled eukaryotes that have the ability to perform photosynthesis. Now, our plant lineages formed by endocytosis, where one of those ancestral cells that had already picked up a mitochondria then went on and picked up a chloroplast. So that's how we get our plants and our algae. But there are some some of our cells, our eukaryotic cells, actually became photosynthetic by a secondary process, a second process known as secondary endosymbiosis. In this process, we see the same thing happen. We see one organism pulling in another, and that organism forming an endosymbiotic relationship. But in our lineages that did not come directly from that primary endocytosis line, the one where a cell engulfed a cyanobacterium, in some of our lineages, like in our Excavata lineage, in our dinoflagellate lineage, these are lineages where some of the organisms acquired the ability to do photosynthesis. And this happened when one protist or one eukaryote engulfed another eukaryote. So we've got three cells here. We have a green algae cell, we've got a red algae cell who themselves are eukaryotes, and then we've got a larger organism. This is a general organism that is a euglena, something that we looked at in lab. It is also a eukaryotic organism because it's got a nucleus and it's got a mitochondria, so we know that it is eukaryotic. And this organism feeds by phagocytosis. So it brings in to a groove, a special groove for feeding. It's basically kind of like a funnel. It feeds by bringing in large particles like a bacteria or a smaller protist, and it engulfs them. Now, eventually, if the cell membrane breaks down, we have this plastid that is acquired by secondary endosymbiosis. So it already has a double membrane because the algae cell acquired the cyanobacteria. Then it had a membrane from the algae cell pulling it in, and now it's got more membranes than that, potentially 3 membranes, because it was engulfed again. So we've got this eukaryote engulfing a eukaryote, and now we have, as evolution moves on, we have this plastid that has been engulfed multiple times, essentially, sequentially. And so what happens is we get organisms outside of the plant lineage that can perform photosynthesis, like these diatoms here. The first supergroup that we're going to cover are called the Excavata group, or the excavates. So Excavata would be the group name, excavate would be the name that we give the individual organisms. And the organism we saw on the previous slide was actually an excavate with that feeding groove. They are called that because of that feeding group. It is as if some of the cell was excavated or dug out. That's what it looks like, so that's where it got its name. And they are heterotrophs that bring in their food energy from phagocytosis. So phagocytosis is the top type of endocytosis in which organisms are bringing in food. There's a couple of different types of endocytosis, but we'll just stick with phagocytosis now. This is how endosymbiosis, whether it's primary or secondary endosymbiosis, occurred. So this includes the group that we call the euglena, or the euglenoids. They are flagellated organisms that move with flagella, and some of them contain those photosynthetic plastids that we talked about that were acquired by secondary endocytosis. There are a couple of common organisms in this group that cause human diseases, in other words, common human parasites. One of them is Trichomonas vaginalis, that is a sexually transmitted infection. And then we've also got Giardia intestinalis. This is an organism that causes intestinal issues like diarrhea, and generally is contracted by drinking contaminated water or contaminated food. So our excavates, we saw a picture already, have this groove in them. The top is called a Jacoba, and it is a general type of euglena. You can see the flagellum on the back, and you can see this groove. Now this groove kind of acts like a funnel to get smaller cells into the organism, and once they come in contact with the inside of this groove, it's going to pinch in, form that little vesicle around the food particle, whatever that food particle might be. Now, if this was endocytosis or secondary endocytosis, this membrane on the outside would become a membrane of that organelle. But this is just showing a bacteria coming in. We could imagine that this is how the cyanobacteria entered that ancient archaea. Or we can imagine this is how a smaller protist entered a larger one and became part of an endosymbiotic relationship. But if that doesn't happen, as is most of the time, that bacterial cell or other small cell is going to get broken down, broken apart by digestive enzymes in the cell. On the right here, we have a euglena, which is commonly a photosynthetic organism that develops its photosynthesis ability from secondary endocytosis. Next, we have the supergroup Archaeaplastida. These are the plants and their relatives. So these protists that come out of the green algae phylum are going to be more closely related to land plants than to any other of the photosynthetic organism. The Archaeaplastida are going to be a monophyletic group. So they evolved from the green algae lineage. The green algae would have been a organism from the ancestral archaeal line that acquired a mitochondria and then acquired the chloroplast. There are three major groups. We're only going to talk about two of them here. The phylum Chlorophyta is the green algae group. They are going to be closely related to our land plants. And when we look at some algae in lab, you'll probably recognize some similarities in the cells. And then we've got phylum Rhodophyta. These are the red algae. Really, the main difference between red and green algae is the type of pigment that they have. So they appear a little bit more red. The last one, the Cryptophyta, we aren't really going to talk about those too much. As far as names that you need to know any of them that I talk about. So we are going to talk about Chlorophyta, we are going to talk about Rhodophyta, but we're not going to talk about Cryptophyta. So you need to know the Chlorophyta and the Rhodophyta, but not necessarily the Cryptophyta. So our green algae occur in freshwater, in the ocean, and occasionally on the land. But if they are found on land, it's because they are in a very damp environment. They don't do well outside of water. So they can be phytoplankton. These are photosynthetic plankton in the water column. They can be periphyton, which are algae that are attached to a substrate like a rock in the shore, or seaweeds, which are attached to the ocean bottom. They are responsible for algal blooms. So every once in a while, algae basically has a population explosion, often due to eutrophication of nitrogen or phosphorus from agricultural runoff, but it can be from other influx of chemicals as well. They are really great as the primary producers in aquatic environments. So heterotrophs in the environment will quite often eat algae. There are only small regions in any particular body of water in which algae can survive because they need sunlight. So they need to live in a shallow enough location where the sun can penetrate and they can harvest that light energy. Most of the green algae are going to be photosynthetic. Some of them contain different types of pigments. So they may appear slightly more green, slightly more yellow, slightly more orange, slightly red, depending on, the type of pigment that they contain, and also depending on that's about it, depending on the pigment they contain. Plastids, as I mentioned, are a general category, so not all plastids contain chlorophyll, but all chloroplasts are plastids. So they just have different types of pigment that can absorb different colors of light more efficiently and reflect different colors of light. So they appear to be something other than just plain green. Our phylum Rhodophyta are red algae. These are usually what we would consider a seaweed. There are also kelps that are a different group. So your brown algae kelp is not the same group as your red or green algae. They have special photosynthetic pigments that allow them to harvest light at much greater depths. So light doesn't penetrate water too well, but it will do it for a while. And then after that, a lot of it doesn't make it very far down. So they have to have really efficient pigments that can collect as much light energy as possible so that they can keep their organism alive. They do not have flagella, and they are generally sessile, which means they cannot move. And so in order to maintain that sort of lifestyle, they have some really complicated life cycles with reproductive cycles that are really kind of crazy. So we are not going to talk about them. You don't have to know what they are. But just kind of a general characteristic is that they don't have flagella, and that results in some weird lifestyle characteristics.